I will talk about somatid from now on. Somatids are microscopic life forms with a very long history of 150 years. Somatid are a science that has been studied by world-renowned scientists like Liff, Bernard, and Bashang. Now, is somatid a part of institutional science or non-institutional science? In the meantime, Dr. Netsang Nasons named it somatid. However, before that, Dr. Bashang Bechamp referred to somatid by another name, called it Microzyma. In the end, both somatid and Microzyma mean very small, the smallest living organism. Is this really a living organism or not? There has been a lot of debate in the scientific community about that. So far, no one has accurately cultured this somatid. Somatid are now recognized as science and confirmed its DNA genes. However, I started researching and developing this somatid 20 years ago and developed a material called QELBY. Although it has the same name, the Dr. Netsang named it somatid. Dr. Bechamp called it Microzyma, and I named this smallest life form that I extracted from ore, and also from plants, QELBY. QELBY is an abbreviation for quantum energy living body. It literally means quantum energy. Living organism. In other words, it's a quantum super microorganism. To be precise, both Dr. Netsang and Bashang Nasons Bechamp conducted genetic analyses, but no one has yet been able to confirm the results of this somatid as an organism through culturing experiments, repeated more than 3 times. So I conducted culturing experiments at Seoul National University using the QELBY I developed. During the cultivation process, we performed high-temperature and high-pressure sterilization in an autoclave twice, and after that, we confirmed the presence of 54 beneficial bacteria. In the cultivation experiment, so this QELBY was extracted from the ore. But is this QELBY a single type? No, it contains 54 types of beneficial bacteria. This is a scientifically astonishing fact, and its significance is very great. However, QELBY generates and emits electrons as a characteristic in itself. When electrons are generated, bioelectricity, similar to the bioelectricity in our bodies, is produced. This somatid generates and emits microcurrent bioelectricity on its own. However, when electrons are generated, electricity is produced. When electricity is generated, oxygen is created. Naturally, aerobic organisms will be activated. Cancer cells, on the other hand, die. Anaerobic harmful bacteria are killed, but aerobic beneficial bacteria, which are activated in the presence of oxygen, proliferate. In other words, QELBY identified 54 types of microorganisms. In his cultivation experiments, and they are indeed living organisms. Is it a living organism or not? In that culture experiment, no one succeeded in the cultivation. And to be clear, QELBY is a somatid and an organic life form, a super microorganism that does not die even after being heat-treated at 1000 degrees for 10 hours. And QELBY exhibits physiological activity effects. According to various experimental results, when QELBY is introduced into water and left submerged for more than an hour, the structure of the water changes. The H₃O water molecules with hydrogen ions to form a new structure, H₃O₂, which is what Professor Pollack refers to as ease water. When the water transforms, QELBY releases electrons and microcurrents into the water. And then absorbs UV light at the 270 nanometer peak. It also emits fluorescent ultraviolet light at the 370 nanometer peak. It emits infrared rays with a much shorter wavelength and higher density than far infrared rays. The scientific principle that QELBY changes. The structure of water to convert it into eased water is clear. In other words, when QELBY is treated with water, the negative electrons and fine electricity it emits along with the effects of UV, and IR infrared rays lead to the transformation into eased water, activating new structures similar to our human cells. That can bond with hydrogen ions. I have conducted many experiments with this water for 20 years. However, QELBY continuously generates oxygen in the water. The amount of oxygen increases. The second change is the introduction of QELBY, which increases the negative ion count in the altered eased water. It also generates voltage and current. The electricity in the water is the same as that. The electricity in the water is the same as that in the human body. And the eased water converted by QELBY acts as a generator itself. In that case, the ORP of the water increases, which means it prevents the oxidation of the water and keeps it alive for a long time. Then, when this QELBY changed, eased water is injected into breast cancer cells. The proliferation of the breast cancer cells is suppressed, leading to their death. This effect has been confirmed in experiments with gastric cancer cells, colon cancer cells, breast cancer cells, and uterine cancer cells, all of which showed inhibition of cancer cell proliferation. Therefore, it has already been published in underground papers. On the other hand, if QELBY is injected into immune cells, it activates the proliferation of immune cells. When viruses or bacteria invade from outside, our body activates macrophages to fight the viruses, which then stimulates the proliferation of immune macrophages. It also activates the proliferation of NK cells that kill cancer cells. Ultimately, this enhances the function of white blood in the blood, resulting in an increased immune response. It's also very important. Yes, reactive oxygen species, which cause inflammation and genetic mutations, need to be removed. Reactive oxygen species are removed, and they help facilitate ATP function, playing a role in recharging bioelectricity. The experimental results regarding these various physiological active functions have been scientifically validated, confirming the inhibition of cancer cells, immune enhancement, the removal of reactive oxygen species, prevention of DNA, gene modification, and the causes of cancer inflammation, such as ROS. These causal factors fundamentally eliminate the causes of cancer inflammation, inhibit cancer cells, and activate immunity. The experimental results regarding these physiological active functions have been published in scientific journals. Therefore, somatids have now transitioned from being non-institutional to institutional science. Based on patented technology, substances are produced, and these substances have been registered in the INCI, International Cosmetic Ingredient Dictionary, for use in the global industry. After manufacturing cosmetics and completing safety tests with the U.S. FDA, they have been registered as medical devices and OTC drugs, establishing a solid foundation for industrialization that can be utilized in daily life. QELBY is now a crucial core substance in the future bioindustry. Thank you.